Hello and welcome back to IB Bio Made Simple. We are talking about topic 11.3, the kidney and osmoregulation. This is part 2 of the notes for this section. So in this video, we are going to be talking about understandings 5 through 8, and we are going to be talking about applications 1 through 3. So those are the things that we're going to be covering today in IB Biology topic 11.3. So last time we talked a lot about the structure of the kidney, and we started talking about some of the structures in the nephron, which can be seen up in the corner here. Specifically, we talked about Bowman's capsule, which can be seen right there in that image, and ultrafiltration, the process by which the blood is initially filtered. All of the chemicals, sugars, amino acids, vitamins, all of those are taken out of the blood, so we can really pick and choose what we want to be left in the blood. So from Bowman's capsule, we are now in the proximal convoluted tubule. You can see that there. So I have my two different tube systems here. In this red line, I can see those are my blood vessels, and in this yellowish orangish line, that is my filtrate. That was what was taken out of the blood. So I have two separate things here. I have filtrate and I have blood. So in the proximal convoluted tubule, the PCT, this is where I'm going to selectively reabsorb some of the most important things. So I do not want to excrete glucose. I do not want to excrete amino acids, vitamins, hormones. I need all those in my body. Those are not waste. So they're going to be reabsorbed here, along with many mineral ions and water. So I have my PCT cell here. On one side of the PCT cell, I have my filtrate, which has all the things I took out of the blood in ultrafiltration. So you should be able to list the chemicals that are taken out of the blood in ultrafiltration. That's one of the things we ended on last time. And on the other side of the PCT cell, I have a capillary that has my blood, which at this point, after it's been ultrafiltered, is essentially just plasma and blood cells and some large proteins. So of course I have microvilli lining the cell because as we know from our study of digestion in topic 6, the microvilli are there to increase the surface area to improve absorption. There's also carrier proteins and some pumps, so things that I want to be in my blood are going to be taken out of the filtrate in through the PCT cell and back into the capillary. That requires a lot of energy, so naturally a PCT cell is going to have many mitochondria, so it can make as much ATP as possible to facilitate that active transport. So to recap, in the proximal convoluted tubule, I have the filtrate, and I am reabsorbing contents that I want back into the blood. It goes from the lumen of the nephron through the PCT cell and back into the capillary. Due to really high concentrations of recovered substances, most of the substances can passively diffuse back into the bloodstream. So because I took everything out of the blood, that means there's really concentrated chemicals in the filtrate. So most of them will move from an area of high concentration here, move up back to the capillary where there's an area of low concentration. So they will happily, passively diffuse. In the PCT cell, I do need to have some active transport though. Some things are too large and will not easily be able to passively diffuse. So things like amino acids, things like hormones, things like vitamins, those are actively transported. Glucose also actively transported in symport with sodium. So what that means is as sodium passively diffuses, because sodium is a small ion, so it will passively diffuse, glucose is coupled to that, so that's where I get symport. So glucose will move across with the help of sodium. And then water is going to follow by osmosis, and we all know osmosis, the diffusion of water from an area of low solute concentration to an area of high solute concentration across a partially permeable membrane. So the water is just going to follow the movement. As all these substances move towards the blood, water is going to follow them. Once I leave the proximal convoluted tubule, I am now in what is known as the loop of Henle. And the loop of Henle, the job here is to control the water and solute concentrations. So this is the part of the nephron that directly controls the concentration of water and the solute concentration. What does that mean for you? Well, it determines how dark your urine is going to be. If you have a lot of excess water, then it's going to leave a lot of water in here, and that's going to dilute the urine, and it's going to look lighter. Whereas if you don't have a lot of water in your system, it's going to absorb as much water as possible, and the urine is going to appear darker. So the job of the loop of Henle is to first generate a high concentration of solutes in the tissue fluid of the medulla. So again, we are dipping below this dotted line here, which means we are now in the medulla area of the kidney, no longer in the cortex. And so it generates a high concentration of solutes in the tissue compared to the filtrate in the nephron. And this helps in the reabsorption of water because, again, water is going to move towards a high concentration of solutes. I also do have some osmoregulation happening in the collecting tube as a result of just some final regulation, so making sure that the water concentration is proper, and that is directly impacted by the hormone ADH, which is anti-diuretic hormone. The loop of Henle generates a high concentration of solutes in the cells and fluid of the medulla. So how does that happen? First of all, I have the filtrate is coming in on the left side, it goes down and then back up the tube. The descending side of the loop over here, this left side, is permeable to water. That means water can move through it easily, but it is impermeable to sodium. The ascending loop is the opposite. It is impermeable to water, but it is permeable to sodium. So the overall effects of that are that the filtrate volume overall, the volume of fluid moving through these tubules, is going to decrease. It also means the output is going to be slightly more dilute than what went in. So what comes out and goes to the collecting duct is going to be slightly more dilute than the filtrate that came in. And there's also a large amount of salts removed in this process. So sodium is pumped into the medulla by active transport. So it is actively removing sodium. Sodium is a positive ion. So because it's positive, it's going to attract negative ions. So chloride ions are also going to follow that. They're going to leave the loop of Henle and they're going to go into the tissue of the medulla. So it is generating that high concentration of solutes. The solute concentration in the filtrate decreases as the loop ascends. So what that means is as my fluid moves up my tube, I am constantly removing solutes. So that means the further I get up the tube, the more solutes have been removed, so the concentration is going to be decreased because I have removed more solutes. And I have a countercurrent effect here that is also facilitating the changes in the loop of Henle. So what that means is I am taking all these salts and pumping them out into the medulla, and as that solute concentration in the medulla is increasing as a result of the activity in the ascending loop, that is going to affect what's happening in the descending loop. So now there's a lot of solutes in the medulla. Water, as we know, moves towards high solute concentrations. So water is going to be moving out. So as the filtrate moves down the descending loop, the solute concentration on this side is going to increase. So as the fluid moves down, more and more water is being pulled out, and that causes the solute concentration to increase because I am losing water, losing volume, but have the same amount of solutes in the descending loop. And so my solute concentration is going to increase. And then it's going to come back around and go through the processes we discussed over here. Those solutes are then going to be pulled out on this side, and it's a recurring cycle. So in this way, I'm able to remove a large amount of salts. And because I remove so many of those salts over here, even though I've removed water here, because I remove so many salts on this side, the output ends up being more dilute than the input. So from there, what's left in the filtrate goes to a collecting duct. And the collecting duct's job is to do a final balancing of water concentration of the blood. So I have my filtrate moving into the duct, moving down, And so it's coming out of the ascending loop of Henle. It went through the distal convoluted tubule and now is in the collecting duct. And it's moving towards the pelvis of the kidney so that it can be transported to the bladder to wait for excretion. So this is the final opportunity for the body to affect things. So as I have that moving from top to bottom, I have blood on the other side that's moving from bottom to top. Water is going to move from the collecting duct into the capillary by osmosis. So when there's a low concentration, or when there's a higher solute concentration here than there is in the duct, water is going to move towards the capillary. They flow in opposite directions, which maintains a counter-current system. That means as the fluid is moving from the top of the duct to the bottom, the blood is moving from the bottom to the top. So water moves from the duct into the capillary and then is taken away. So new blood is then exposed to what's in the duct and it can continue to pull water. That counter-current system prevents these two ducts from ever being equalized. Because they are moving in opposite directions, they will never become equalized, so there will always be a drive to pull water back into the blood and try to excrete as little water as possible. If a person is dehydrated, so if they have not consumed enough water, they do not have enough water in their system, your body releases ADH, which is antidiuretic hormone. And that acts on the walls of that collecting duct and makes it more permeable to water. So if a person is dehydrated, then more water is going to leave their duct and enter back into their capillary. So they are going to reabsorb more water, they're going to excrete less water, and that is going to mean that the urine output is hypertonic, has a really high solute concentration, because I am making it really concentrated by removing the water. Osmoregulation is an example of negative feedback control that we talked about previously. This is using hormones for negative feedback control. So when there's not enough water, I release a hormone that helps me maintain water. And as that water in the blood increases as a result of pulling it out of the urine, then it's going to decrease the need for ADH. So because the result decreases the stimulus here, that is a negative feedback. This is a really important point. Water conservation in animals, because all animals, of course, need water. So what happens is we see a correlation between the length of the loop of Henle and water conservation. So for instance, there's this rat, this kangaroo rat. Its loop of Henle is very long. Because it lives in deserts, it needs to conserve as much water as possible. So a longer loop of Henle allows it to do so. Kangaroo rats can concentrate their urine to 3,500 millimoles per liter, whereas humans can only concentrate it to 400 millimoles per liter, so almost 10 times more concentrated, because they need to retain as much water as possible due to the fact that they live in the desert. So what I see here, I have a graph here with relative renal medullary area on the bottom, so a larger medulla as I go towards the right, and a maximum urine osmotic concentration. So how concentrated can I make my urine over here? And the size of the medulla is a direct result of the size of the loop of Henle. So all these indicators are different animals, and the shape tells you what group of animal they belong to. So you can see here that as I move to the right on the x-axis, as my loop of Henle gets longer, they are able to more highly concentrate their urine. So that length of the loop of Henle is directly correlated to the level of concentration to which animals can get their urine, and therefore the amount of water that they can conserve within their body. They can excrete much less water, which is beneficial if you're in an environment where water conservation is really important. So an IB question you could be asked is to explain osmoregulation in the kidney. This would be an 8-mark question, one of the ones at the end of paper 2. So take a minute here, pause the video, and see if you can jot down some notes. How would you answer a question like this? And then your response could look something like this. Osmoregulation has to do with the loop of Henle, so there's a couple different notes you could talk about there. and the collecting duct. So there's a couple different notes that you can talk about there. So hopefully you hit as many of those as possible. If you didn't, make sure you go back and review. What are the applications of this? We always have to talk about applications. So consequences of dehydration. We all know that dehydration is a risk, but also overhydration is a risk as well. So dehydration, as we all know, is when the body does not have enough water. So some results of that are thirst, obviously, smaller quantities of darker urine. You'll start to feel some lethargy, so some exhaustion, lower blood pressure, increased heart rate because of the lower blood pressure, an inability to regulate body temperature, and in severe cases, seizure, brain damage, and death. So all of these are, of course, going to be more dangerous the hotter the temperature gets. So sometimes we'll see examples of athletes, for instance, getting heat stroke because they are dehydrated and they have all of these symptoms, and the heat makes it much worse, and that can be really dangerous. Overhydration, however, is also a problem. You can have too much water. You need to have a lot of water to have too much water, and you really kind of have to be trying to do it, but it is possible to have too much water. Clear urine is, of course, a symptom, but you can also have, which is dangerous, the swelling of cells due to osmosis. Because you have so much water in your body, that water will move into the cells. That can cause headache and disruption of nerve function due to swelled cells, and in more serious cases can cause delirium, blurred vision, seizures, coma, and death. So it's really important, we can see here, to maintain proper hydration. So especially if you are working out in adverse conditions, whether it's really hot or really cold, you need to be cognizant of your water consumption. We also have treatment of kidney failure. Because the kidneys are so important, kidney failure can be very dangerous. So treatment of kidney failure focuses on two main approaches, which is hemodialysis, which is what you see here. This is a dialysis machine, and also kidney transplants. So kidney failure is what happens when the kidneys can no longer adequately filter the blood. And what can happen there is that you get a buildup in toxins, you get a buildup in nitrogenous waste in the blood, and that can cause damage to the cells. And if not treated, kidney failure can be very serious and can lead to death. Dialysis is the most common treatment for kidney failure. Dialysis treatment lasts about 4 hours and is done a few times per week. The number of times it's done depends on the severity of the kidney disease. So essentially, a dialysis machine works just like your kidney. Blood is removed from the patient. It is filtered, and then it is returned to the body. So it's basically an external kidney. It uses semi-permeable membranes to get rid of small particles like urea, but retain larger particles. And so it can act like an external kidney, but it does not suffice for a permanent solution. In many cases, as the kidneys continue to deteriorate, there is a greater and greater need for dialysis, and eventually even a dialysis machine cannot overcome the risk of kidney failure. So for a long-term treatment, the best solution is a kidney transplant. Donors are either going to be someone who has recently died and is an organ donor, or it can be a person who has chosen to give up one of their 2 kidneys. You do not need 2 kidneys to live, you only need 1 healthy kidney to live. So a donor can donate one of their healthy kidneys, and then both the donor and the person who receives the donation would both have 1 healthy kidney. Interesting thing about kidney transplant is that they don't actually remove the old kidneys. It's actually not really worth the risk because these blood vessels are so large and very important. It's not really worth the risk of dealing with them. There's no damage to the body to be had by having those kidneys in the body. So they just graft the new kidney into the lower abdomen with the renal artery, renal vein, and ureter connected to the appropriate places, and it functions just fine. Then the person will have 3 kidneys in their body, but importantly, they'll have 1 functional one. And finally, we have urinalysis. So diagnostic tests of urine provide an easy way to test illness and drug abuse. There are many, many things that can be tested in the urine because the body is taking out toxins. Toxins can be things like some drugs, so they can be detected in urine samples. But you can also test other things. So, for instance, we said that you do not want to have any glucose in your urine because you want to maintain that. If I found glucose in urine, that could tell me that there's an indicator of diabetes because glucose is not being properly regulated. You can detect some blood cell infections, some diseases, some cancers. If there are trace amounts of proteins, that can indicate kidney disease because proteins should not be in the urine. So all of these things can be tested and many more. Urine tests are a quick and easy way to judge the health of a patient, and that's why they're done pretty routinely. They're the most common way of drug testing in professional sports, and also most times you go to a doctor, they'll just take a quick urine sample because they can get a lot of information very quickly just from urine. That concludes our 11.3 notes. If you have more questions, I would strongly suggest you go look up BioNinja. They do a fantastic job of walking through all of these topics. I hope you learned a lot. If you have any questions, please leave them in the comments and we will answer them as quickly as we can. Thank you for watching, and I'll see you next time.